The study I will be presenting today is titled Allogenic Immunity Clears Latent Virus Following Allogenic Stem Cell Transplantation in SIV-Infected ART-Suppressed Macaques. Before we get started, let's define a few things and break down that title a little bit. SIV stands for Simian Immunodeficiency Virus, which is very similar to HIV, but this infects non-human primates such as monkeys or gorillas. So for these experiments, they studied SIV-infected macaques, which is a genus of primates, and more specifically, they studied the cynomolgus macaques. ART stands for antiretroviral therapy, and it is a combination of medications given to those with HIV that reduces the amount of HIV in an individual's body. This is called viral suppression, and it can make the viral load so low that it is undetectable. CCR5 is a protein on the surface of white blood cells that is involved in immune response. However, some people have a mutation of this CCR5 protein called delta 32, and people with this mutation are resistant to HIV infection. Viral reserves refers to a group of immune system cells that are infected with the virus but are inactive or latent. These are resistant to antiretroviral therapy and can become reactivated if it is discontinued. Allogenic simply means taken from different individuals of the same species. So in this case, they are transplanting allogenic stem cells from monkeys with immunity to SIV. In other words, those monkeys have the delta 32 mutation on the CCR5 protein, and they are giving them to the SIV-infected monkeys. Lastly, before we move on, we have engraftment. This is when the blood-forming cells an individual receives during a transplant start to grow and make new healthy blood cells. So what was the purpose of this study? Well, although researchers have recently discovered that allogenic stem cell transplant from donors with the delta 32 mutation of CCR5 can cure HIV, the mechanisms, or how it works, has still been unclear. The three theories were that either 1, the pre-transplant immune conditioning essentially removed the latent viral reservoir, 2, the donor cell-mediated allogenic immunity eliminated the inactive infected cells, or 3, the new CCR5 mutation of the individual's immune system made it resistant to HIV rebound events. And through these experiments, they were able to demonstrate that it is mostly allogenic immunity that was able to destroy the latent viral reserves of the SIV infection in some cases. So for their experimental methods, they again used the cynomolgus macaques, or colloquially known as the crab-eating macaques. They picked this species as the model because they accurately model HIV infection and immunity. So first, they infected 8 of these monkeys with SIV and then initiated daily ART. By day 35, they had an undetectable viral load, and after 9 to 17 weeks of having this undetectable viral load, 4 of the monkeys underwent an RIC regimen, a reduced intensity conditioning, followed by allogenic stem cell transplant. They were then tapered off of the ART between day 57 and 132 days post-transplant. The other 4 monkeys remained on ART only and acted as the controls to this experiment. Long-term successful engraftment was achieved in all 4 of the macaques that received the allogenic stem cell transplant, as the researchers were able to see in the neutrophil, lymphocyte, and platelet reconstitution. This was stable through 600 days post-transplant. They measured this by monitoring donor chimerism. Now, chimerism just means a single organism with more than one cell genotype. In other words, they measured the donor-mediated replacement of the recipient's T cells. And after extensive measurement of donor chimerism from blood samples and biopsies of lymph nodes, spleen, colon, bone marrow, and other areas, they found a high level of donor-derived cells, from 75 to 100%, with the vast majority of immune cell populations being over 90% donor-derived. They also found that in 3 of the primates, the CD4 T cells were 95 to 100% donor-derived. However, one of the primates only reached 90% donor-derived CD4 T cells. In one primate that had the highest level of donor CD4 T cells, SIV was completely undetectable and was thought to be completely eradicated. In the next primate, they found very low levels of SIV in the blood and lymph nodes, levels that were detectable but under the limit of quantification. In the third primate, only 1 lymph node had detectable levels of SIV DNA. Finally, the last primate that had the lowest levels of CD4 T cell replacement had the highest levels of SIV detected. The researchers then did further experiments to validate their findings. These results suggest that the primary mechanism of viral reserve eradication after stem cell transplantation is allogenic immunity, which is defined as the replacement of recipient CD4 T cells with donor CD4 T cells. I think these findings are extremely promising, and I am so relieved to see progress being made towards finding a cure for HIV, especially given the devastating history of HIV and AIDS. Due to the stigma of AIDS in the early 1980s, there was little effort to stop the epidemic and an extremely delayed response in funding research into the disease. To this day, over 500,000 people die yearly from AIDS, and the research is still considered to be underfunded. But with the recent discovery of allogenic immunity, we are closer than ever before to putting an end to this terrible virus. I hope you enjoyed this presentation. Thank you for watching.